children who had genetic diseases. So I spent a lot of time learning about mutations. And I'd been taught in school that mutations were primarily haphazard. And what I found was that this largely wasn't true. What we learned in school is that your body is controlled by genes. Genes are made up of DNA. They're inherited according to Mendel's laws, dominant, recessive. And genes code for proteins, which do the work of the cell. Genes change by mutations, which is changes in the DNA sequence. And we were taught that mutations are caused by copying errors and other sorts of accidental sources, and that most mutations are bad, but the good ones are kept through natural selection. The problem with this is that the mutation rate is so high that if mutations were primarily accidental, then natural selection couldn't keep up, and life would degenerate by collecting bad mutations that couldn't be removed. So where did we get this idea that mutations were random? Well, first, Mendel discovered the gene, and then we discovered mutations which modify genes. Then we learned that external changes, like cutting tails off mice, were not heritable, but that radiation caused changes that were heritable but arbitrary. And so this led to the random mutation hypothesis that says that mutations are random with respect to the needs of the organism. And this influenced several generations of biologists. But this was all before the DNA revolution. When we learned what genes were made of, that they were made of DNA, we learned that it's the sequence of DNA that codes for proteins. And so when you combine the idea of random mutation with DNA sequences, it led to the expectation that there'd be lots of accidental sequence changes that would cause leftover junk DNA that would just kind of accumulate in your cells. And so when biologists began to learn about different types of genetic materials such as mobile DNA, repetitive DNA, and pseudogenes, which look like genes but don't code for proteins, they assumed that this was all evolutionary junk that was left over from random mutations. But in the last few decades, we've learned more and more about the cell, and we've realized that it's a tightly integrated system of nanomachines that are all working together in a system. So are mutations perhaps part of this system? Well, we learned several interesting things. First of all, we learned that mutations can be targeted. Most mutations occur in tissue-specific genes and not in the housekeeping genes that keep the cell running. And the tissue-specific genes are the ones that are the precise ones that need to be changed as the environment changes. We also learned that mutations can be induced. If you grow E. coli and starve it from its normal sugar and grow it in the presence of a different kind of sugar, a specific DNA sequence will be inserted. Mobile DNA will come in and be put in the right place to allow E. coli to adapt. In your body, you produce millions of antibodies to combat infections by antigens. But there's only a few hundred genes that code for these millions of antibodies. Well, how does this work? The answer is cut and paste DNA. So these few hundred genes are actually gene parts, and they're grouped into buckets. And the antibody cell pulls a part from each bucket and combines it into a final gene. And so by recombining these parts over and over again, you can create millions of antibodies. Then when an antigen comes in, the body finds the closest matching antibody cell and mutates its gene to make a closer fit. So now your genome is 3 billion base pairs long, and the antibody gene is only 1,200 base pairs. All of the mutations go to the antibody. Not only that, they go to the right parts of the antibody. They go to the part that attaches to the antigen. Another type of mutational process is repeat DNA, which acts as tuning knobs for cell functions and body structures. In dogs, a single repeat segment will vary the bend in its nose. Pseudogenes are used as templates for creating new genes out of old genes. And although they don't code for proteins themselves, cells can take modules from pseudogenes and use them to create variations in other genes. So what we see is that the cell, the mutation process, is actually, rather than haphazard, it's actually informationally directed. And so your cell is pre-coded with information to tell the cell how to mutate. And so what I learned is that just as hearts have heart attacks, so the mutation process, designed to help life adapt, can also have diseases, but they are the exception rather than the rule. Thank you.